going to talk about biology of aging. So the last 100 years have seen amazing advances in human health and an enormous increase in human lifespan, at least in the developed world. And we have a great example, illustration of that here. This is Madame Jeanne Calment, who died at the age of 122 years in 1997. So she's the current record holder for longevity. And she was mentally quite clear right into her last years. And she remembered seeing Vincent van Gogh in her hometown of Arles when she was a little girl, just to give an idea of her amazing longevity. So there's a lot to celebrate in terms of our human advances in health leading to increased lifespan. But there's a downside to this, which is, of course, as people are living longer and longer, they're surviving long enough to experience illnesses like this, Alzheimer's disease. And this is an English writer, Iris Murdoch, who tragically was struck down with Alzheimer's disease. And you can see her here just before she dies, and she's really a shell of a human being. You can see in her eyes that she's gone mentally. For that reason, Alzheimer's disease is sometimes called a living bereavement. The body lives, but the mind is gone. And Alzheimer's is one of the many diseases that are associated with aging, and you don't need to read through this here. This is just a list of some of the things that happen to our bodies as we age, including cardiovascular disease, so the diseases that give rise to stroke and heart attacks, and the many cancers that are associated with aging. So the point that I'd like to make is that all of these diseases are produced by a biological process, which is the aging process, the biology of aging. And really, at the moment, in the developed world, most of the disease and illness that people experience is the result of aging. Aging causes disease. It causes diseases of aging. Therefore, it's very important to understand the biology of aging. It's surprising that until relatively recently, biologists haven't put a great deal of effort into understanding it. But before I go on to the biology of aging, I'd like to say something kind of controversial, which is to criticize the way that aging-related diseases are currently treated in terms of as problems for medical research. So here are some of the major diseases which are largely caused by aging. Cancer is largely a disease caused by aging. So each one of these diseases has its own distinct research field. So you have people who are interested in cancer, working in the field of cancer research, and they have their own journals, their own institutes, and so on. People working on cardiovascular disease have their own field of cardiovascular disease research. And the result of this is that treatments arising within each field tend to be specific to the disease which is being treated. So if you have a new treatment for cancer, the chances are it's not going to be useful for treating diabetes. But as I've said, these diseases are really all symptoms of the same thing, which is aging. So surely a much better approach, a more effective approach, would be to target aging. If you could have a treatment for aging, then you have potentially something that would protect against all of these diseases. A little like bacterial infections. So in the old days, individual bacterial diseases were treated in distinctive ways, and then antibiotics were developed that could protect against the broad spectrum of bacterial diseases. Now, some of you might look at this and think, well, that sounds nice in principle, but surely this is a fantasy, this is a dream. But people working on the biology of aging think otherwise, because in the laboratory you can achieve this. And here's an example which Maria Basco mentioned, dietary restriction, or sometimes called caloric restriction. It's been known since the 1930s that reducing the food intake in rodents, like this rat here, can slow aging and slow all of the aging-related diseases that rodents get. For example, cancer. You can greatly reduce cancer levels in elderly rodents by caloric restriction. So really the point that I'm making here is that if you really want to cure diseases of aging in a powerful way, if you want particularly to find treatments for those really difficult to treat diseases like Alzheimer's disease, really the key is to understand the biology of aging. So what is aging? I think it's fair to say that at the moment, in a profound way, we don't know what aging is. There are various mechanisms that are emerging that seem to contribute to it, but at a profound level, we still don't know what aging is. But one basic fact about aging is clear from this little figure here. This shows the maximum lifespans of 3 mammals, and you can see how very different it is. And when you think about it, it's quite strange because each of these organisms is made of the same basic stuff. They're made out of skin and blood and bone, and they have the same basic internal organs, but they age at very, very different rates. And what this tells us is something very, very fundamental about aging, which is that, like many other features of living organisms, it's controlled by genes, like your eye color, your body size. Your aging rate is very much controlled by your genes. Therefore, it's interesting to ask what particular genes control aging. Biologists have studied the way that genes control characteristics for a century. And there's a standard approach which you can use to this, which is sometimes called the classical genetic approach. And that involves working with mutants, with strains where you've changed one individual gene. And in this case, mutants with altered aging rates. If you could identify mutants with altered aging rates, then you ought to be able to solve aging through the following strategy. You map and you clone and you sequence the gene. You find what the gene is that's been changed. You find out what the gene does, what sort of protein, what sort of biochemical processes it alters, and that should lead you to an understanding of aging. So it all hangs on whether one can find mutants with altered aging rate. Can you make animals live longer by changing them genetically? So a very good organism for doing this is this creature here. This is C. elegans. It's a worm. It's about 1 millimeter long as an adult. Biologists use C. elegans to try to solve very complex questions about biology, because this is a very simple organism, and if you can understand something in this organism, then perhaps you can then understand it in human beings too. And the great thing about C. elegans in terms of aging is its lifespan is only 2 or 3 weeks. They grow old and die after only 2 or 3 weeks. So you can study the genetics of aging very easily in this animal in a way that would be very difficult in humans. So here is an example of a long-lived mutant. And the discovery of this mutant was a real exciting moment in the biology of aging. So here's a plot. What this shows here is 2 populations of worms. A wild-type population, a normal population in blue, and a mutant population in green. And here, on the y-axis, this shows you the percentage of animals that are alive, and here's time in days on the x-axis. So initially, all the animals are alive, 100% are alive. And if we look at the blue curve, after about 20 days, the animals are dying of old age, and gradually they're all dead by about 30 days. And here in green is another different strain of worms with a mutation in a gene called DAF2, And you can see their lifespan is doubled. So it's very interesting to ask, well, is DAF2 a gene that exists in people? Because many of the genes in the worm are the same genes that control growth and development and so on in humans. And it turns out that this gene encodes a protein which is present in humans. It's a protein which is involved in pathways that are involved in sensing the nutrient state and control growth. So this is the insulin and IGF receptors. So the discovery of this long-lived mutant led to the identification of a pathway that controls aging, a system that regulates the aging rate. And it's a system that is sensitive to the level of nutrition. This is a simplified version of the system. So here's the receptor, the insulin receptor, and this is the surface of the cell. And it controls a cascade of kinases, and then they switch off this protein here, which is a transcription factor called DAF16. So if you reduce the activity of this pathway, you activate this transcription factor, and that produces this big increase in lifespan. So in a way, this transcription factor is what's really interesting. This is the gene that controls directly the aging process. And just to show you, this is an amazing a piece of data that came out from a group in Arkansas in the US last year. And this is another mutation affecting this longevity control pathway in the worm. And here you see another survival curve for wild-type worms. And here are two different mutants. And those mutants live 10 times longer than the wild-type. So that just shows you the power of these nutrient-sensing pathways to control aging. A 10-fold increase in lifespan. It's incredible. So genes really have very, very powerful effects on aging. So really the challenge, something which many people are working on, including my lab, is to try to understand how this increase in lifespan is generated. And really we've discovered this control system that regulates aging, but what we know is that it works through this transcription factor. And the transcription factor is a protein that controls other genes, you see, and here are the genes that the transcription factor controls. And the assumption is that the genes that are regulated by DAF16 are the processes that directly control aging. And in a sense, this is a sort of like a holy grail within the field, is to discover what these processes are. Because I think if we can understand what processes produce magically this 10-fold increase in lifespan, probably at that point we'll understand what aging is, at least for the worm. And if we can understand aging in the worm, then maybe we can understand aging in humans too. So I'm going to tell you one story about our efforts in my lab to try to understand how these long-lived worms live long. And that was to say, well, there is a theory of aging which has been around for many years, which is basically a theory that says that aging is the result of oxidation. It's like when the butter goes rancid, it oxidizes. I mean, the air that we breathe, that we live in, is really a very caustic gas, and it burns and oxidizes many things. So in the same way that metals rust, the theory goes, biological systems oxidize, and that's what causes aging. So here's a picture of the theory. The theory particularly views these things. This is a mitochondrion, which is the part of the cell that generates energy. And the mitochondria can produce free radicals, particularly a form of oxygen called superoxide, which is very, very reactive. And the idea is that this reactive free radical from the mitochondria goes around the cell, damaging the constituents of the cell, and that's why we age. So this is a very influential theory, and it's why if you go into health food stores, you'll see bottles of antioxidants with tablets that you can take that potentially are supposed to slow your aging process. So the cell has a defense mechanism against this particular reactive oxygen species, and it's an enzyme called superoxide dismutase, which I'll call SOD for short. So this is an enzyme that gets rid of this reactive oxygen. So potentially this is an enzyme that should protect against aging, according to this oxidative damage theory. So when these long-lived mutants were first discovered, and this was back in the early 90s, one of the first things that people said was, well, the reason they're probably long-lived is because they have resistance to oxidative damage, because that's what the theory says. And if that's true, then these mutants should have more of this enzyme, this antioxidant enzyme, SOD. So they looked, and they found, sure enough, there was more SOD in these worms. And they said, well, that probably explains it then. That explains why they're long-lived. But is it? What they showed is a correlation between the level of antioxidant enzymes and longevity. But does the increase in this antioxidant enzyme actually cause the increase in lifespan? That hadn't been shown. So that's something that we've been investigating for the last few years, for the last 5 or 6 years. And basically, what we've done is to manipulate the levels of this antioxidant enzyme in the worm. And these are the antioxidant enzyme genes, the SOD genes. There are 5 of them. And to see whether they're determinants of aging. So we can ask, if we knock these genes out, if we take the genes away, remove the antioxidant defense, do the worms age faster? If we overexpress the antioxidant enzymes, do they live longer? And we can also ask, is the increase in lifespan in these long-lived mutants caused by the increase in this SOD enzyme? And just to give you one bit of data from our studies, this one of the things we did was to knock out the SOD from the mitochondria. And there are 2 genes that produce the mitochondrial SOD. So the mitochondria is where this superoxide radical is particularly produced. And this just shows you the survival of worms. These are in blue, the worms that lack the SOD in the mitochondria. And this is paraquat and hyperoxia. These are 2 forms of oxidative stress. So we're increasing the oxidative stress, and the worms live shorter. So they are hypersensitive to oxidative stress. So they have a real problem with oxidative stress, as you'd expect. So the question is, what effect is there on aging of removing these antioxidant genes? So here we see survival curves for wild type in black, and these are 2 mutants, each missing one of the 2 SOD genes, and their lifespan is normal. So we then looked at worms lacking both of the SOD genes, and their lifespan is normal as well. So there's no effect on aging. And this was a big surprise, because this went completely against the oxidative damage theory. So to cut a long story short, I'll just give you the conclusions. We manipulated the antioxidant defenses in the worms in many ways, and we saw very little or no effect on aging. We also showed that the longevity of these DAF2 mutants is not due to the increased resistance to oxidation. So it would seem in worms, at least, aging is not caused by oxidation. Meanwhile, in other labs, for example, looking at mice, a number of similar studies were performed overexpressing antioxidant enzymes in mice, and the lives were not extended. And also studies have come out looking at the effect of dietary antioxidants on people, and showing that there's no increase in lifespan. And in fact, in some cases, mortality is actually increased in people taking antioxidant supplements. So the theory really seems to be incorrect. So if the theory is wrong, what's the truth then? So I'd like to present 3 alternative models about sort of beyond this oxidative damage theory. One model, which I call the neo-oxidative damage theory, this says that maybe the theory could still be true. And maybe it's simply that the forms of reactive oxygen that we were looking at, which was particularly superoxide and hydrogen peroxide, they're not the most important forms of oxidation. Maybe there are other kinds of oxidation that are important. But I don't really believe that theory, because there are other forms of molecular damage that are known to be very important in aging. For example, reaction between sugar and molecules of the body. It's called glycation. It's very important in aging. So an alternative theory is to say, well, the oxidative damage theory wasn't wrong, but it was a half-truth. It was right insofar as damage to the molecules that make up our bodies causes aging. But it was wrong to say that it's purely oxidation that's important, that maybe a much broader spectrum of forms of molecular damage cause aging. So here, this replaces the oxidative damage theory with what I call the molecular damage theory. But there's a third possibility which is very interesting to consider, which is that maybe aging isn't caused by damage at all, primarily. We know that damage is associated with aging. We see more damage during aging, but maybe aging causes the damage. Maybe it's not the damage that causes aging. And this we call the supramolecular dysfunction theory. Okay, at this point I'm going to, for the rest of my talk, I'm going to talk about something slightly different and tell you about a kind of a leap forward, I think, that's happened in the biology of aging in the last few months, which I think is very exciting. But let me say first, I think in a way there are 2 aims of the research at the Institute of Healthy Aging and 2 goals within biogerontology. And in a way, the main goal, we're dealing with scientists after all, they want to understand aging. They want to know what aging is. But there's a second goal, which is to try to improve the health in older people. And my view has always been that we have to understand aging first, and then the other thing will follow. But some of the recent findings are making me change my mind and think that maybe, even without understanding aging fully, we can do something to help this old lady here, or to help all of us, in fact. So let me go back to this phenomenon of dietary restriction or caloric restriction. One idea that people have discussed for years, sometimes in the bar after scientific meetings, is the possibility of developing drugs that would produce the same effect as dietary restriction. Dietary restriction is very difficult for people to do. The level of reduction of food intake is so extreme that for most people it's intolerable. But maybe if you could have a drug that would produce the same effects, that would be very helpful. And how helpful this would be was underscored by I'm going to talk about 3 papers that have just come out that kind of go together, I think. And this is one of them. This is a paper that showed, after a study that took something like 25 years, that caloric restriction actually works in primates. So these are rhesus monkeys, and their lifespan is up to 40 years. So the experiment is still ongoing. It started in the late 1980s, but here you can see this is a one monkey that's been on a full diet, and here's one of the monkeys that's been subjected to dietary restriction. And you can see here the survival curves. Here are the fully fed monkeys, and here are the dietary restricted monkeys. And the health status, as you can see here, these monkeys are healthier. You can see the fur is better, the skin is less wrinkled, you can see here. So this raises the likelihood that caloric restriction would work in humans. Now let me come back to the worms and the insulin pathway. So one of the things that I've been involved in over the years is to try to see whether genes that control aging in the worm control aging in other organisms. And an early study which we conducted at UCL, this was done in collaboration with Linda Partridge at UCL, was to ask whether this pathway that produces these large effects on aging in worms also has similar effects in a fruit fly. And that's very important because it was possible that these lifespan effects might be peculiar to nematodes. We wanted to know whether they would work in humans, but that's obviously very difficult. But if the pathway controlled aging in fruit flies and worms, which are evolutionarily very separated, then it makes it much more likely that, that this pathway would control aging in mammals too. And on the back of our findings from fruit flies, we got funding to study this question in mice, but we were scooped by Martin Holzenberger in particular, who showed that if you knock out the IGF-1 receptor gene, which is really the same gene as DAF2, you see an increase in lifespan in mice. And we did carry on doing these experiments, and in 2008, and this is work very much from Dominic Wither's lab, we found that the same mutation that produces this increase, the same gene where mutation increases lifespan here in flies, which is a gene in this insulin pathway, also increases lifespan in mice. And moreover, these mice stay younger for longer. So all of the diseases of aging seem to be slowed in these mice. So in terms of trying to bring together studies of dietary restriction and these mutant studies, there's a kind of an overall model, which is that which is that these pathways are functioning to monitor levels of nutrients in order that organisms can make the choice between either growth and reproduction on the one hand or survival on the other hand. And we've recently been broadening our studies to the whole network of genes that respond to nutrients and control growth and longevity. And we've particularly started working on this pathway here, which is often called the TOR pathway, and acts through a kinase called ribosomal S6 kinase here, which I'm going to talk about now. So it's been shown in worms and flies that the effects of dietary restriction actually involve this TOR pathway. Okay? So the TOR pathway seems to be how dietary restriction produces effects on aging in worms and flies. But in the mouse, this wasn't known. So what we did, or this is work that was particularly in Dominic Wither's lab, was to look at the S6 kinase mutant mice. So this is this gene in the TOR pathway. And what we find is that if you knock this gene out, the mice live longer. It's about a 20% increase in mean lifespan. And this effect is actually seen in females, but not in males. And that's quite common with these mutants, that the effects are often seen in females, but not males. So let me tell you a little bit more about these mice. They're very interesting mice. First of all of the measures of aging-related disease that we looked at were slowed down in these mice. So they stay younger for longer. They're not living longer in a moribund state. Their youth span is extended. Secondly, very interesting, they look just like mice under dietary restriction. They're lean, their body size is small, their glucose homeostasis is better, and the gene expression patterns look very similar to those of wild-type mice under dietary restriction. They also show elevated levels of an enzyme called AMP-activated kinase, or AMP kinase for short. And the gene expression patterns look like the expression patterns of mice treated with a drug that activates AMP kinase. So here's AMP kinase. It's a trimeric kinase, and it's a kinase which is switched on by energy stress. So if the available energy in the cell drops, this kinase is switched on. It lowers anabolism, growth-related processes, and it increases energy availability. So this suggested the idea that perhaps the activation of this enzyme in these mice was what was actually making them live longer. And there was a precedent for that, which is in worms, these long-lived insulin IGF mutants that I told you about, and also some forms of dietary restriction. The increase in lifespan requires the presence of this enzyme. So we have clues that this enzyme is very important in promoting longevity and slowing aging. So we tested this idea in worms. We used a mutant worm lacking S6 kinase. It's a gene called RSKS1. And here are wild-type worms in blue. And in red here is the S6 kinase mutant. It's long-lived. Not a huge effect, but it's there. It's significant. And if we look in a mutant called AAK2, which lacks the AMP kinase, the longevity is gone. These S6 kinase mutants also have other defects. They're small, they grow slowly, their fertility is reduced. And in each case, the defect is dependent on this AMP kinase. So this suggests a model, which is this is very exciting because the model is that the dietary restriction is working through the TOR pathway. That's what this data is implying. And the AMP kinase activation is part of the longevity program. And the reason why this is so exciting is that this pathway is druggable, right? If you can drug this pathway, in principle, you should have a dietary restriction mimetic. In other words, a drug that would slow aging, would recapitulate the effects of caloric restriction without having to reduce your food intake, slow aging and extend lifespan. Okay, so this is very exciting. So that's why I suddenly realized maybe we don't understand how the pathway works, but maybe we can drug it and get benefits that could be really of use to people. So the implication then is that if you were to inhibit mTOR or S6 kinase with a drug or activate AMP kinase, then you would slow aging. And it so happens, and this is the third of the 3 papers, that it's just been shown in July of this year that if you treat mice with this drug called rapamycin, which is an inhibitor of TOR, it slows aging. Moreover, AMP kinase has a number of activators that are well characterized, particularly a drug called metformin, which is used widely for the treatment of type 2 diabetes. So the model predicts that metformin activating AMP kinase should increase lifespan. And this is data, unpublished data from my lab, showing that metformin increases lifespan very well in C. elegans. And there's also some studies from a group in St. Petersburg showing that at least in short-lived mice that metformin also increases lifespan. So I think that with the appearance of these 3 studies together, all of a sudden we seem to be much closer to actual treatments that could be used preventatively to reduce aging-related disease in general and improve health in older people. So maybe we can really produce something that could help this old lady here, and us all, of course. And just to say thanks to the people, particularly the postdocs who did the bulk of the work here. And thank you for listening.